Welcome back to the Deep Dive. We're here to take that complex stack of sources you sent overall about the human blueprint, its glitches, and how we've evolved, and really pull out the core insights you need.
It's quite a stack today.
It really is.
Yeah.
We're charting basically the entire map of human biological diversity. We're going from Mendel's simple rules of inheritance right up to how humans adapt physiologically to, some pretty extreme environments.
Yeah, it's a huge scope. We have to zoom right in, down to the genes, and then zoom way out to see how populations cope with deserts, high altitudes, cold, you name it. Our sources hit genetics fundamentals, what happens when that genetic blueprint breaks.
Which can be pretty drastic.
Absolutely. And also the tools we actually use to track all this variation across populations.
So our mission today really is to make sense of all this dense material, give you a clear structure. We want you to walk away not just with definitions, but a real connected understanding the biological gears that define us and also make us so diverse.
Mm-hmm. And that gets us to the big question, doesn't it? The kind of paradox at the heart of it all.
Which is?
How do these really basic, simple rules, figured out by Mendel with his pea plants, of all things, lead to this incredible, mind-boggling complexity and diversity we see worldwide, and maybe even more crucial for health? What happens when those rules just completely break down, like at the chromosome level?
Okay, so let's dive into that inheritance architecture. Where do we start?
Got to start with Mendel.
Right, Gregor Mendel. It's still kind of amazing, isn't it, that experiments with pea plants back in the 19th century still give us the basic math, the framework for understanding how humans inherit traits. We're talking his three main laws,
Exactly, that's the enduring power here. So first up, the law of segregation. This is, it's about randomness, basically. It says when you make gametes, sperm or eggs, the two copies, the alleles you have for each gene, they separate. So each sperm or egg only gets one of those alleles. This split ensures genetic variety, And it's fundamental for figuring out how traits pass down and lets us predict probabilities. Really important for family studies, risk calculation.
And then things get a bit more complex with the law of independent assortment. This one deals with multiple traits at the same time.
That's right. It says that the alleles for different genes separate independently of each other when those gametes are forming. Now, the big caveat, the important point, is this only really holds true if the genes are on different chromosomes. Or if they're on the same chromosome but really far apart, so they're effectively unlinked.
So if you're looking at, say, inheriting earlobe shape and maybe blood type, if they're on separate chromosomes, the outcome for one doesn't influence the other.
Precisely. And the third one, the law of dominance. This is crucial for what we actually see, the phenotype. If you're heterozygous, meaning you have two different alleles for a gene.
Right. One dominant, one recessive.
Exactly. That dominant allele will completely cover up or mask the effect of the recessive one in your observable traits.
Okay, so those are the foundational rules. How do researchers actually apply these to messy human families? That's where pedigrees come in.
Pedigrees, yes. Pedigree construction. It's much more than just drawing squares for males and circles for females, though that's the start. Genetic counselors use these visual maps, these family trees, to track traits, calculate exact risks, and even identify people who might be carriers for a disease without knowing it.
Because the pattern gives it away.
The pattern tells you so much almost immediately. Take autosomal dominant traits, think Huntington's disease or Marfan syndrome. They usually show up in every generation. Why? Because you only need one copy of that dominant allele to express the trait.
Makes sense.
But then look at autosomal recessive traits like cystic fibrosis. These can seem to skip generations. They only appear when two carrier parents, who often look perfectly healthy themselves.
Right, they each have one recessive copy.
Exactly. They both pass on that recessive allele to their child. So the child gets two copies, and then the trait shows up.
And then you've got the sex chromosomes involved with X-linked inheritance. Things like hemophilia.
Hemophilia, Duchenne, muscular dystrophy. Yeah, those are classics. Because males only have one X chromosome, they're hemizygous for genes on the X. They're much more vulnerable to X-linked recessive conditions. If they inherit that one affected X from their mother, who's usually a carrier, they'll have the condition.
Okay. This really highlights the difference between traits controlled by just one gene versus, most human traits, right?
Absolutely. It forces us to draw a really clear line between monogenic or single-factor inheritance and multifactorial or polygenic inheritance.
Monogenic seems straightforward enough. One gene follows those Mendelian patterns we just talked about. Cystic fibrosis, Huntington's, hemophilia, classic examples. The risk is pretty predictable if you know the parent's genes.
Right, but and this is a huge but, most of the variation you actually see walking down the street, that falls under multifactorial or polygenic inheritance. This is where it gets complicated. We're talking multiple genes, maybe dozens, maybe hundreds,
Wow.
all interacting with each other and with environmental factors, big-time environmental influence.
So this is stuff like height or BMI, maybe even intelligence or risk for diseases like diabetes or heart disease.
Exactly, all those things. You don't get a simple yes or no outcome. You get a whole spectrum, a continuous range of variation across the population.
Okay.
Statistically, it usually forms that classic bell-shaped curve.
And how does that work with multiple genes? Is it just more genes equals more trait?
That's the core idea, yeah. The concept of additive effects, it's cumulative. The more positive alleles that push the trait in a certain direction you inherit, the more strongly that trait is expressed. So your final height isn't down to one tall gene, it's the combined effect of maybe hundreds of genes, each adding a tiny bit, plus your nutrition, your health during childhood. It all adds up.
Okay, that makes sense. But let's circle back to the real world of studying human genetics. It can't always be that neat, even for the single gene stuff. What are the complications, the limitations researchers run into? Where does the blueprint get messy?
Oh, it definitely gets messy. A big one is incomplete penetrance. This is huge. It means someone can have the known disease-causing mutation, the faulty gene sequence confirmed by testing, but show absolutely zero symptoms, no sign of the disease whatsoever. Mendry model assumes 100% penetrance. If you have the gene, you show the trait. But in reality, especially in humans, you have to account for this. It really complicates risk assessment in families.
And even if the gene is expressed, it's not always the same way, is it?
Exactly. That's variable expressivity. You and I could have the exact same mutation for, say, a dominant condition, but the severity of my symptoms could be way different from yours. And the range of symptoms, which specific problems show up, can vary wildly too. So even with a confirmed genetic diagnosis, predicting the actual clinical outcome for a specific person, really challenging.
And then there's the issue of things looking genetic when they're not.
Yes, crucial point, phenocopies. These are individuals who show symptoms, a clinical picture, that perfectly mimics a known genetic disorder. But the cause isn't genetic at all. It's environmental.
Like, I remember the thalidomide tragedy. Babies born with severe limb defects. That looked like some rare genetic syndrome. That's right. But it was caused by the drug exposure during pregnancy.
A devastating and perfect example. So a key job for geneticists and counselors is figuring out, is this pattern we're seeing in a family truly inherited, or is it a phenocopy caused by some shared environmental exposure? It's a critical distinction.
Okay, so gene expression can be incomplete, variable, or even mimicked. But what about when the genes themselves are just bad news, the ones that cause serious problems or are lethal?
Right, the ultimate biological cost. We talk about lethal and sublethal genes.
Lethal genes, these cause death, right? Or really severe developmental problems. You mentioned Huntington's earlier.
Yeah.
Even though symptoms show up later, it's considered a dominant lethal.
Genetically, yes, because having just one copy of that mutated allele inevitably leads to fatal neurodegeneration down the line.
Wow.
But here's something fascinating from the sources, maybe a bit unsettling. Almost every single one of us walking around carries multiple, potentially lethal recessive mutations hidden in our genome.
Really? Hidden or how?
They're masked by our functional dominant allele, the healthy copy. But this hidden reservoir of harmful mutations, it's called the genetic load. It's present in every population, contributing to the baseline genetic risk.
Okay, so that's the lethal load. What about sublethal?
Sublethal or semi-lethal genes, these are definitely harmful, but not immediately fatal. Hemophilia is a great example again. The impaired blood clotting isn't instantly deadly in itself.
Right.
But what would normally be a minor cut or even internal bruising can suddenly become life-threatening because the clotting factors aren't working properly.
And there's one more category, conditional lethality.
Conditional lethality. This is super interesting. The gene itself is only lethal under specific circumstances, specific environmental triggers or physiological conditions. Like G6PD deficiency, common enzyme issue. Having the gene variant isn't deadly on its own. But if someone with G6PD deficiency is exposed to certain oxidative stressors, specific drugs like some antimalarials, or even eating fava beans, it can trigger massive destruction of red blood cells, hemolytic anemia, which can be fatal.
Wow. So the environment is the switch.
Exactly. Or PKU, phenylketonuria. It's a metabolic disorder. Untreated, the buildup of phenylalanine is toxic to the brain, leading to severe intellectual disability, effectively lethal in terms of normal development. But with a strict diet started right after birth, avoiding phenylalanine, the person can live a completely normal life. It's the ultimate gene-environment interaction story.
Okay, so we've covered single genes, their variations, their complications. But what happens when the problem isn't just one gene, but the entire structure, the chromosomes themselves? Humans have 46, right?
Yeah.
22 pairs of autosomes, 1 pair of sex chromosomes, neatly organized.
Usually neatly organized. But chromosomal aberrations are when that organization breaks down. These are big changes in the number of chromosomes or in their physical structure. They disrupt the genetic balance dramatically because you're suddenly changing the dosage of hundreds, maybe thousands, of genes all at once.
And that leads to developmental issues, syndromes.
Often very severe developmental syndromes, yes. The dosage imbalance is just too much.
What causes these errors in number?
The main culprit for numerical errors is a failure in cell division called non-disjunction. It's a mechanical error, basically. During meiosis, the process that makes sperm and eggs, either the homologous chromosomes in meiosis the first or the sister chromatids in meiosis the nemetal, fail to separate properly. They don't get pulled apart like they should.
So what happens then?
You end up producing gametes, sperm, or eggs that have the wrong number of chromosomes, either one too many or one too few. This state is called aneuploidy.
Are there known risk factors for non-disjunction?
Yes, the sources are clear on this. The most well-known factor is advanced maternal age, especially over 35.
Why is that?
It's thought to be because a woman's eggs are arrested in meiosis from before she's even born, potentially for decades. As those egg cells age, the cellular machinery responsible for accurately separating the chromosomes, the spindle fibers, might become less efficient, less reliable. This increases the chance of a chromosome pair failing to separate correctly. Other potential factors mentioned include radiation exposure or maybe delayed fertilization after ovulation, but age is the big one.
Okay, so non-disjunction leads to the wrong number. How are these numerical errors categorized?
Well, first there's polyploidy. This is extreme. It means having extra entire sets of chromosomes, like triploidy, having 69 chromosomes in total, 3 sets, or tetraploidy, 92 chromosomes, 4 sets.
Wow. Can humans survive that?
Almost never. It's incredibly rare in live births and generally incompatible with life. These usually result in very early miscarriages.
So the more common scenario is?
Aneuploidy. That's having the wrong number of individual chromosomes, not whole sets. The most common type of aneuploidy that results in live births is trisomy, having 1 extra chromosome, so 47 total instead of 46.
And the classic example everyone knows is Down syndrome, right? That's trisomy 21.
Exactly, trisomy 21. It's the most common trisomy that allows survival after birth, occurring in about 1 in 800 live births.
Why is trisomy 21 more viable than other trisomies?
It comes down to gene dosage, essentially. Chromosome 21 is actually one of our smallest chromosomes. It carries fewer genes compared to, say, chromosome 13 or chromosome 18.
Oh, okay.
So having an extra copy of 21, while it causes the features of Down syndrome, the overall genetic imbalance is less severe, less disruptive to development than having an extra copy of a larger chromosome packed with more genes. That's why trisomy 13, Patau syndrome, about 1 in 5,000 births, and trisomy 18, Edwards syndrome, 1 in 6,500, are far, far more severe.
They have profound effects.
Profound organ defects, yes. Survival beyond the first few weeks or months is unfortunately very rare. The gene dosage imbalance from those larger extra chromosomes is just overwhelming for the developing system.
We also see aneuploidy involving the sex chromosomes, right? Those seem to be relatively common.
They are, and they often have less severe effects than autosomal trisomies, partly because of X inactivation. These are the sex chromosomal aberrations.
Like Klinefelter syndrome, that's XXY.
Yes, Klinefelter syndrome, 47XXY. It affects males about 1 in 1,000 male births. The extra X chromosome leads to certain features, often tall stature, maybe longer limbs, sometimes reduced facial hair, gynecomastia, some breast development, and critically, they are usually infertile because the testes don't develop properly.
And for females, there's Turner syndrome. That's missing an X.
Right, Turner syndrome, 45X. It's a monosomy, only 1 X chromosome instead of 2. Affects about 1 in 2,500 female births. Key features often include short stature, maybe a webbed neck appearance, sometimes heart defects, and importantly, the ovaries don't develop fully, leading to infertility. That second X chromosome is needed for normal female development and fertility.
And there's also triple X.
Triple X syndrome, 47XXX, affects females, about 1 in 1,000 births. Interestingly, many individuals with triple X have few or no distinctive physical features. Fertility is usually normal. There might be a slightly increased risk of learning difficulties or delayed speech development, but often it goes undiagnosed. It shows how effectively the body can shut down extra X chromosomes through inactivation, reducing the impact of that extra dose.
Okay, so that covers errors in chromosome number.
Yeah.
But the structure can also break, right? Rearrangements.
Absolutely. Structural abnormalities. These are large-scale rearrangements of the chromosome material, even if the total count might still be 46.
Like what kinds of rearrangement?
One common type is translocation. This is when a piece of one chromosome breaks off and sticks onto a different, non-homologous chromosome.
Okay.
In a reciprocal translocation, two different chromosomes just swap pieces. If no genetic material is lost or gained in the swap, the person carrying it is usually healthy. They have all the right genes, just in a slightly different order. They're called a balanced carrier.
But there's always a but, isn't there?
There's often a but in genetics. The problem comes when they make sperm or eggs. Because of the rearranged chromosomes, they have a high risk of producing unbalanced gametes with missing or extra pieces of chromosomes. This can lead to infertility, miscarriages, or children with birth defects.
And there's a special type of translocation, Robertsonian.
Robertsonian translocation. Yes, this is specific. It only happens with the acrocentric chromosomes. Those are 13, 14, 15, 21, and 22. They have one very long arm, the Q arm, and one really tiny short arm, the P arm. In a Robertsonian translocation, the long arms of two different acrocentric chromosomes fuse together at the centromere. The two tiny short arms are usually lost.
Lost. Is that bad?
Usually not. Those short arms mainly contain genes for ribosomal RNA, and we have lots of copies elsewhere, so losing them is generally okay. The person ends up with only 45 chromosomes, but they have essentially all the important genetic material. They are typically healthy carriers.
But again, problems making gametes.
Again, yes. High risk of producing unbalanced gametes, which can lead to conditions like translocation Down syndrome if chromosome 21 is involved, or Patau syndrome if 13 is involved.
Okay, besides translocations, what other structural changes happen? Deletions.
Deletions, yes. Loss of a chromosome segment, it's a partial monosomy, can be from the end, terminal, or the middle, interstitial. The classic example is Cri-du-Chat syndrome. That's caused by a deletion on the short arm of chromosome 5. It leads to severe intellectual disability, other physical features, and that distinctive high-pitched cat-like cry in infants.
And the opposite of deletion.
Duplication. Having an extra copy of a chromosome segment, a partial trisomy. Large duplications usually cause problems, leading to developmental syndromes. But, interestingly, small duplications are thought to be really important in evolution.
How so?
They provide extra genetic material, raw material that can then mutate and potentially evolve new functions over time without messing up the original gene's job. A source of new genes, essentially.
Fascinating. Any other rearrangements?
We also see inversions. A piece of the chromosome breaks off, flips around 180 degrees, and then reattaches.
Flips around.
Yep. If the break doesn't include the centromere, it's paracentric. If it does include the centromere, it's pericentric. Like balanced translocations, inversions often don't cause problems for the person carrying them, as all the genes are still there.
But meiosis again.
Meiosis again. The inverted segment causes problems when homologous chromosomes try to pair up. They have to form awkward loops to align properly. This can lead to issues during crossing over, resulting in unbalanced gametes with deletions and duplications.
Okay, this is getting complex. And there's one more layer. Mixoploidy.
Right, mixoploidy. This means an individual has more than one genetically distinct cell line within their body. Two main types.
What's her?
Mosaicism. This is when you have two or more cell lines that originated from a single zygote, but a mutation or chromosomal error happened after fertilization during early development. So some cells are normal, some have the error. The effects depend on which tissues are affected and how early the error occurred.
And the other type?
Chimerism. This is much rarer and kind of mind-blowing. A chimera is an individual composed of cells derived from two different zygotes that somehow fuse together very early in development.
Two different fertilized eggs fusing into one person.
Exactly. Essentially, two individuals merged into one. It can happen, for example, with fraternal twins fusing in the womb. Remember that case, Lydia Fairchild? DNA testing showed the kids she gave birth to weren't genetically hers based on her cheek swab DNA.
I think I heard about that.
Wow. Turned out, the cells in her ovaries had a different genetic makeup. The genetic makeup of her fused twin than the cells in her cheek. She was a chimera. It just shows the incredible, sometimes baffling complexity underlying even basic biological identity.
Okay, shifting gears a bit. We've talked about the blueprint and when it breaks. Now, how do scientists actually track and measure all this variation across different human populations? This is where genetic markers come in, right?
Exactly. These are the signposts we use to map human diversity. But not just any trait works as a good marker.
What makes a good marker?
Well, first, it has to be clearly genetically determined. Second, it should have a relatively simple, understandable inheritance pattern, ideally Mendelian. Third, we need to be able to classify it accurately and reliably in individuals. And fourth, crucially, it needs to show common variation polymorphism in the population. If everyone has the same version, it's not useful for tracking differences.
Makes sense. What are some of the classic markers? The source mentioned blood markers a lot.
Yes, the blood markers were among the first and are still incredibly useful because blood is easy to get, and the types are stable genetic traits. The big one, of course, is the ABO system.
Right. AB, and O blood types, determined by alleles on chromosome 9.
That's one. 3 main alleles, A, B, O. Now, A and B are codominant. If you inherit both, you express both, giving you type AB blood. O is recessive to both A and B, so you need 2 O alleles to be type O.
Technically, what are A and B?
It's about sugars on the surface of red blood cells. There's a basic molecule called the H antigen. The A allele codes for an enzyme that adds one type of sugar, and acetylgalactosamine, to H. The B allele codes for an enzyme that adds a different sugar, D-galactose. The O allele is basically a non-functional version. It doesn't add any extra sugar, so you just have the basic H antigen.
Okay, and these vary globally.
Hugely. Type O is the most common worldwide, around 47%. AB is the rarest, maybe 3%. But the fascinating part is the geographical gradient.
How so?
The frequency of the B allele is noticeably higher in central populations, think Central Asia, Northern India. The A allele tends to be more frequent in peripheral populations, Europe, Australia. Type O is pretty common everywhere, but especially high in the Americas.
Why the difference? Just chance.
Probably not just chance. These patterns likely reflect ancient migrations, genetic drift, but also maybe natural selection. There's evidence suggesting different blood types might offer slightly different resistance levels to certain historical infectious diseases, like plague, smallpox, or cholera. That could have driven frequency changes over time.
Interesting. What about the RH factor, positive and negative?
The RH system, yes. Primarily about the D antigen. If you have it, you're Rh positive, Rh plus. If not, Rh negative, Rh. Rh plus is dominant and much more common globally. But again, there's significant variation. Our sources highlight that something like 99% of East Asians are Rh plus E, while the frequency drops to maybe only 60% in some European populations, like the Basques, who have a uniquely high frequency of Rh negative.
So another clear population difference.
A very clear one, yeah. Great marker for population studies.
Beyond the standard blood types, what else? The sources mentioned the HLA system.
HLA. The human leukocyte antigen system. This is incredibly important. It's a group of genes located on chromosome 6.
What does it do?
It's the core of your immune system's ability to tell self from non-self. HLA proteins sit on the surface of your cells and present little fragments of proteins from inside the cell to your immune cells, specifically T cells. If those fragments are foreign, like from a virus or bacteria, the T cells recognize them and mount an attack.
Okay. Why is it a good marker?
Because it is hyperpolymorphic, insanely variable. There are thousands of different versions or alleles for the various HLA genes. This means the combination of HLA types any one person has outside of identical twins is almost unique.
And that's important for
Transplantation. It's the basis of histocompatibility, or tissue matching. The closer the HLA match between organ donor and recipient, the lower the chance of immune rejection. Because it's so variable, it's also an incredibly powerful tool for anthropologists tracing ancient population movements and relationships. The patterns of HLA diversity tell deep stories about human history.
Wow. Okay. Any other protein markers?
Sure. Things like serum proteins, haptoglobins, for instance, they bind to free hemoglobin that gets released when red blood cells break down. There are different types, and some studies suggest certain types might be more common in tropical areas, possibly linked to protection against malaria or other conditions causing hemolysis.
And enzymes. You mentioned G6PD earlier.
Right, G6PD, glucose-phosphate dehydrogenase, protects red blood cells from oxidative damage. Deficiency, as we discussed, makes you sensitive to certain triggers, but also seems to offer some protection against malaria. So, unsurprisingly, the gene variants causing G6PD deficiency are most common in regions where malaria has historically been a major selective pressure. It's a classic example of selection maintaining a potentially harmful allele because it also provides a benefit in a specific environment.
Okay, blood groups, HLA, enzymes.
Uh-huh.
But wait, the sources have a whole bit on earwax. Seriously, how is earwax a genetic marker?
Yes. It sounds trivial, doesn't it? But earwax, or cerumen, is actually a fantastic textbook example of a simple single-gene trait that shows really strong global population differences. It's consistency. It's basically either wet sticky, which is dominant, or dry flaky card, which is recessive. This difference is controlled largely by a single gene, ABCC11.
And the variation.
It's stark. The wet type is overwhelmingly common in people of European and African ancestry. The dry type is overwhelmingly common in East Asians and Native Americans. The frequency difference is so dramatic, it's one of the clearest single-gene markers distinguishing these broad population groups. It tracks ancient migration routes out of Africa incredibly well.
That is genuinely surprising. Earwax tells a story of human migration.
It absolutely does. And it's not the only seemingly minor trait like that. Take PTC tasters, the ability to taste a bitter chemical called phenylthiocarbamide.
I think I remember doing that in a biology class. Some people taste it strongly, some taste nothing.
Exactly. And that ability is largely controlled by a single gene, TAS2R38, with the tasting allele being dominant. Again, big frequency differences. Tasters are more common among populations with African and East Asian ancestry. Non-tasters are more common among Europeans, and interestingly, very common among Australian Aboriginal populations, another marker reflecting population history and isolation.
Amazing. One more. BAIB.
BAIB excretion, beta-aminosobutyric acid. It's an amino acid, and some people excrete large amounts of it in their urine high excretors. It seems to be related to muscle turnover or breakdown. Again, follow simple genetics, and being a high excretor is quite rare in Europeans, but much more common in people of East Asian ancestry. So whether it's a complex system like HLA or something as seemingly mundane as earwax, these genetically determined variable traits are the crucial tools for mapping human diversity.
Okay, let's zoom out again. From genes and markers to how human biology actually changes, how we adapt in response to the huge range of environments we inhabit. Deserts, Arctic cold, high mountains. But first, the sources for us getting the terminology right. Adaptation isn't just getting used to something short term.
No, the terms are specific and important. Adaptation in the strict biological sense refers to long-term evolutionary change. It's about changes in the genetic makeup of a population over many generations driven by natural selection. Think about the evolution of dark skin pigmentation in populations living near the equator as protection against high UV radiation. That's a genetic adaptation.
Okay, so that's inherited. What about short-term changes?
That's acclimatization. This is a temporary, reversible physiological adjustment made by an individual in response to an immediate environmental stress. Getting a tan in the summer, that's acclimatization. Shivering when you step out into the cold. Acclimatization. Your body adjusts, but those changes aren't passed on to your kids genetically. When the stress goes away, your body generally reverts.
Right, and there are other terms. Plasticity.
Biological plasticity. This is sort of the underlying capacity of an individual organism to change its physiology or morphology in response to environmental cues. Losing weight during a food shortage is an example of plasticity. Your body can change in response to the environment.
Okay, and one more.
Oh.
Developmental.
Developmental response, or developmental acclimatization. These are changes that happen during the critical period of growth and development, and they are irreversible. A classic example is someone who grows up at high altitude. They often develop a larger chest cavity and greater lung volume to cope with the low oxygen. That's a change fixed during development. It won't revert even if they move to sea level later in life.
Got it. Adaptation is genetic, population level. Acclimatization is individual, reversible. Developmental response is individual, irreversible, set during growth.
Nicely summarized. Now, applying this, biology has these classic ecological rules that try to link body form to climate. Allen's rule, Bergmann's rule.
Right. Allen's rule, that's about limbs. Shorter limbs in cold climates to conserve heat, longer limbs in hot climates to radiate heat.
Correct. Minimizing or maximizing surface area relative to volume.
And Bergmann's rule is about overall body size. Bigger, bulkier bodies in colder climates because they have a lower surface area to volume ratio, which helps retain heat. Smaller bodies in warmer climates.
Exactly. Think of a sphere versus a pancake. The sphere holds heat better. So generally, we see trends like stockier builds in Arctic populations compared to more linear builds in many equatorial groups.
And there's one for skin color. Gloger's rule.
Gloger's rule. Generally, darker pigmentation is found in populations from more humid, tropical environments closer to the equator, likely driven by the need for UV protection. Lighter pigmentation is found in populations farther from the equator, especially in less sunny or more arid regions, potentially related to balancing UV protection with the need to synthesize vitamin D in the skin.
Okay, so these rules describe general patterns linking body form to environment, presumably through long-term adaptation. But how well do they actually apply to modern humans? The sources seemed a bit critical.
They are critical, and rightly so. While these rules describe potential evolutionary pressures, and you can see general trends that align with them across indigenous populations, for modern, globally dispersed humans, culture is king. Meaning, our cultural buffers, technology, clothing, housing, heating, air conditioning, global food transport, have massively reduced the direct selective pressure of climate on our physical form. We don't need shorter limbs to survive in the Arctic anymore. We have incredibly effective parkas and heated buildings. Our primary adaptation now is cultural and technological, not purely biological. These rules are interesting historical context, but they don't dictate modern human variation in the same way.
That makes sense. We adapt the environment to us as much as adapting to it. Let's take a specific example. Heat stress, like in a hot desert. How does the body cope physiologically?
Okay, hot desert. The main goal is keeping that core body temperature stable, somewhere between roughly 35 and 41 degrees Celsius. Above that, you risk heat stroke.
How does it cool down?
First, vasodilation. Blood vessels near the skin surface widen, bringing more warm blood closer to the surface to radiate heat away. But if the air temperature is actually hotter than your skin temperature, radiation doesn't work. You might even gain heat.
So then what?
Then it's all about sweating. Evaporation is incredibly effective at cooling. As sweat evaporates from your skin, it takes a lot of heat with it. In extreme heat, evaporation can account for 90% or even more of your body's heat loss.
But that costs a lot of water, right?
A huge amount. You can lose a liter, maybe even more, of fluid per hour through sweat in really hot, dry conditions. And you're not just losing water. You're losing electrolytes, salts, like sodium and chloride. Dehydration and electrolyte imbalance are major risks.
So how does acclimatization help here, getting used to the heat?
Acclimatization makes the whole sweating process more efficient and less costly. After repeated heat exposure over days or weeks, several things happen. You start sweating sooner. At a lower core temperature, you sweat more. But crucially, the concentration of salt in your sweat decreases significantly.
So you cool effectively, but conserve precious electrolytes.
Exactly. Your cardiovascular system also adapts. Your heart rate doesn't spike as high for the same workload. Studies on groups who live in hot climates like Bedouins or Kalahari Bushmen show these physiological efficiencies compared to unacclimatized people.
But again, biology isn't the whole story. Humans use culture here too, right?
Massively. Cultural adaptations to heat are critical. We create a more manageable microclimate. Think about traditional desert architecture.
Like adobe houses in the American Southwest, or those thick-walled buildings in the Middle East.
Perfect examples. Materials like adobe, stone, thick mud brick, they have high thermal mass. They absorb the intense heat during the day very slowly, keeping the inside relatively cool. Then at night, they slowly release that stored heat, keeping the inside warmer when the desert air gets cold. Compact building designs also minimize the surface area exposed to the sun. Some groups even use subterranean dwellings.
And clothing makes a difference too. You'd think less is more in the heat.
Not necessarily. Think about traditional desert clothing, like the loose, slate-colored robes worn by Tuareg or Bedouin people.
Right, covers everything. Exactly. Light colors reflect solar radiation. The looseness allows air to circulate, promoting evaporation. But critically, the layers actually help maintain a slightly more humid microclimate right next to the skin, which can sometimes slow down the rate of water loss through sweat while still allowing for effective cooling. Studies have actually shown that appropriately clothed, acclimatized individuals can sometimes maintain thermal balance with less sweating than if they were nude in the same conditions. Culture optimizes the biological response.
So it's a biocultural system. Let's touch on another marker mentioned, hemoglobin levels. How do that relate to environment?
Hemoglobin, HB, the protein in red blood cells that carries oxygen. Levels naturally vary with age highest in newborns, generally increasing until adulthood. And gender males typically have slightly higher levels than females, partly attributed to menstrual blood loss in females.
But environment plays a role.
Yeah.
Altitude.
Altitude is the big one. People living permanently at high altitudes where there's less oxygen in the air generally have higher HB levels. Their bodies produce more red blood cells to maximize oxygen capture and transport. This is a classic physiological adaptation. Or, like among Tibetans, they might have genetic adaptations for more efficient oxygen use, not just higher HB. It shows different adaptive pathways to the same stress. Socioeconomic factors like nutrition also impact HB levels, of course.
And briefly, sensory perception. Does that vary?
Sensory perception definitely changes with age. Vision tends to peak in young adulthood, maybe declining noticeably after 40. Hearing often peaks even earlier, maybe starting to decline subtly after 25, especially for high frequencies. But culture and environment interact here too. Someone working in a loud factory might experience noise-induced hearing loss. Cultural diets profoundly shape taste preferences and maybe even sensitivity. Smell sensitivity can be influenced by occupation or environment too. It's all interconnected.
Okay, this next section is complex. We need to address the concept of race, how it's been defined historically, how science views it now, and the ethical dimensions. The sources provided some historical definitions.
Yes, they did. Historically, especially in early anthropology, race was often seen through an essentialist lens, like Wheaton's definition, viewing races as major divisions of humankind, each sharing a bundle of physical features inherited from a common ancestor, sort of fixed categories based on appearance.
But that view evolved.
It did, moving towards the population concept, heavily influenced by geneticists like Dobzhansky and anthropologists like Montague. This view defines races not as fixed types, but as Mendelian populations groups that interbreed, which differ statistically in the frequencies of certain genes. It shifted the focus from typology to population genetics.
But even that population concept has faced criticism, right? Especially when it comes to applying rigid categories. The sources highlighted the UNESCO statement on race.
Yes, the UNESCO statements, starting in the 1950s, were landmark efforts by scientists to publicly counter pseudoscientific racism. They stressed several key points based on the scientific understanding of the time, which largely holds true today.
What were the main takeaways?
First and foremost, all humans belong to a single species, Homo sapiens, and share a common origin. Critically, they emphasize that so-called racial distinctions based on biology have no connection to national, religious, linguistic, or cultural groups. These are separate dimensions of identity.
And the idea of pure races.
Completely debunked. They stated clearly there's no such thing as a pure race due to the long and constant history of human migration and intermixing. And importantly, they affirmed that mixing between different population groups is not biologically harmful.
And the most crucial point, perhaps.
But there is absolutely no scientific basis for ranking groups in terms of innate mental capacity or ability. Biological differences that exist are unrelated to intelligence or cultural potential. All groups possess the full range of human capabilities.
Okay. Still, it's probably useful to understand the kinds of traits that were used in those older, outdated classification systems, like Montague's categories.
Right. Understanding the history helps understand why it was flawed. Those older systems, like the three major races often cited, Caucasoid, Negroid, Mongoloid, relied almost entirely on a small number of visible morphological traits. Things like skin color, grouping people into broad categories like leucoderms, light skin, xanthoderms, yellowish, melanoderms, dark, hair form, Leotricious, street, Cymatris, wavy, curly, Eulotricious, tightly coiled, woolly, facial features, like the presence or absence of the epicanthic fold, the mongoloid fold in the eye region, and even cranial measures, like the cephalic index, the ratio of head width to head length, classifying skulls as dolichocephalic, long-headed, brachycephalic, broad-headed, etc.
So basically judging populations based on a few external physical traits. What's the main scientific critique of that approach?
The fundamental problem is that these traits massively oversimplify human diversity. First, many of these traits, like skin color, are actually adaptations to local environments, like UV levels. They don't reflect deep overall genetic differences between populations. They reflect specific selective pressures.
Right.
Second, these traits vary continuously, not in discrete jumps. Where do you draw the line between wavy and curly hair or different shades of skin color? The boundaries are arbitrary. Third, different traits don't cluster together neatly. Someone might have dark skin but straight hair, for example. The traits don't always travel as a package deal.
So what's the reality according to modern genetics?
The reality, revealed by studying actual genetic markers like the ABL blood groups, the Rh system, HLA types, even things like fingerprint patterns, dermatoglyphics, is that human variation is clinal. It changes gradually over geographic space.
Clinal, meaning?
Meaning frequencies change gradually, like a slope, rather than having sharp breaks between groups. While we can see differences in the average frequencies of certain alleles between populations, like the high frequency of whorl fingerprints in East Asians versus loops in Europeans, Africans, these are statistical differences, not absolute ones. There are no genes that are present in 100% of one race and 0% of another.
And most genetic variation is found
Within populations, not between them. If you compare the genetic diversity between two individuals from the same town, it's often much greater than the average genetic difference between two widely separated populations. The concept of discrete biological races just doesn't hold up to genetic scrutiny. Modern science talks about population structure, genetic diversity, ancestry, and clines, not rigid racial boxes.
Given that scientific understanding, it's important to define racism clearly as a social construct built on flawed biological premises. How do the sources define it?
The sources define racism neutrally as a system of discrimination, prejudice, or antagonism directed against someone based on the belief that one's own perceived race is superior. It's rooted in those discredited ideas of inherent racial difference and hierarchy.
Yeah, how does it manifest? It's not just individual prejudice.
No, it operates on multiple levels. Institutional racism refers to how biases and discriminatory practices get built into the policies and procedures of organizations like in hiring, housing, healthcare, or the justice system. Systemic racism is broader, referring to how racial inequality becomes embedded within the very structures of society, leading to disparities in wealth, health, education, etc., often without overt individual prejudice being the main driver.
Yeah.
And then there are more direct forms like microaggressions, colorism, prejudice based on skin tone within a group, and racial profiling. It's a complex social system, not just individual attitudes.
Okay. Let's connect this back to genetics in a practical sense. How does this understanding of variation and the potential for misuse inform ethical practices in medicine, like genetic counseling and screening?
It's hugely important. Genetic counseling is a specialized service focused on helping individuals and families understand and cope with genetic information. Counselors use tools like pedigree analysis to assess risk, but their core role is providing information, exploring options, and offering emotional support in a non-directive way, empowering informed choices.
Right. And genetic screening.
Genetic screening is more population-focused, systematically testing groups to identify individuals with specific genetic variations or mutations associated with disease risk. This includes things like newborn screening for metabolic disorders like PKU we discussed, carrier screening for prospective parents, prenatal screening during pregnancy like NIPT, and even pre-implantation genetic diagnosis, PGD, for embryos during IVF.
So what's the ethical bridge here?
The bridge is that both counseling and screening deal with incredibly sensitive personal information, our genetic blueprint. Given the history of misuse of biological concepts like race, there's a profound ethical obligation to ensure this genetic information is handled with extreme care. This means robust protections for privacy and confidentiality, ensuring truly informed consent, and actively guarding against genetic discrimination in areas like insurance or employment. Understanding that human variation is complex and clinal, not categorical, is fundamental to applying these powerful genetic tools equitably and ethically. #outro.
Wow. Okay, that was quite a journey. We started with Mendel's elegant, simple laws of inheritance in pea plants, then navigated the, frankly, often messy and sometimes catastrophic consequences when chromosomes break or rearrange. We explored the toolkit of genetic markers from blood types to earwax, used to track diversity.
We looked at how human bodies and critically human cultures adapt and acclimatize to the planet's extremes, heat, altitude.
And finally, we tackled the really complex history and current understanding of race, emphasizing the scientific view of clinal variation over outdated categories and the ethical imperative that comes with genetic knowledge.
Before we finish, there's one last practical application nugget in the sources that really ties the blueprint idea together: DNA profiling.
Like DNA fingerprinting.
Exactly.
Sure.
Also called DNA typing. It doesn't usually look at genes, but at highly variable, non-coding regions of DNA called STR, short tandem repeats. Everyone has these repeats, but the number of repeats at specific locations varies hugely between individuals, except identical twins.
So you'd create a unique profile.
A unique genetic profile, yes. It's indispensable in forensics for matching samples from crime scenes to suspects, also used routinely for paternity testing and increasingly for tracing ancestry and building family trees and genealogy. It's a direct application of understanding individual genetic variation.
And related to that is gene mapping.
Right. Gene mapping is the broader effort to figure out the exact location of genes on our chromosomes. There's genetic mapping, using inheritance patterns, and physical mapping, using molecular techniques. Locating genes is the essential first step towards understanding their function and identifying the genetic basis of inherited diseases. It's the ongoing project of reading the blueprint in detail.
So this whole deep dive has really been about understanding the human blueprint, its rules, its variations, its vulnerabilities.
Which leads to a final thought. We talked about genetic load, that hidden burden of potentially harmful mutations we all carry.
Yes, that background level of deleterious alleles present in any population, usually masked in heterozygotes, but collectively reducing the average fitness slightly.
And the sources mention that this load can be amplified, made more visible.
It can, particularly through practices that increase homozygosity, meaning increasing the chances of an individual inheriting two copies of the same allele. The most significant way this happens is through consanguineous matings.
Marriages between close relatives, like first cousins. Exactly. Because close relatives are more likely to share the same rare recessive alleles inherited from a common ancestor, their children have a significantly higher chance of inheriting two copies of one of those harmful recessive alleles. This brings those hidden components of the genetic load out into the open, leading to a higher incidence of rare recessive genetic disorders in offspring.
So understanding these fundamental principles, inheritance, mutation, genetic load, the effects of mating patterns, it's not just academic theory.
Not at all. It's profoundly practical. It provides the knowledge base for making informed decisions about personal health, family planning, public health policies, and even understanding the structure and history of human societies. It equips us to engage critically with our own biology.
A powerful toolkit indeed. So the final question for you, listening, now that you have this deepened understanding of your own blueprint, its history, its variations, what will you do with it?